will introduce our special guest today. Dr. Richard Gordon is a group leader in clinical neuroscience at the UQ Centre for Clinical Research and an advanced Queensland Mid-Career Fellow. He is also a science ambassador for the World Parkinson's Coalition and is involved in the promotion of the 6th World Parkinson's Congress to be held in June 2022. Any information that Dr. Gordon provides today is general in nature, and if you did want any specific information relating to anything that he talks about, we do remind you that you should ask your GP or health specialist. So today, Dr. Gordon will be discussing the emergence of the evidence on how changes in the gut microbiome can play a role in Parkinson's disease onset and progression. It will also cover the restoration of gut biome as a potential strategy for diagnosis and treatment of Parkinson's. Finally, the talk will outline how this approach of microbiome restoration for Parkinson's will be evaluated in the Combo PD clinical trial led by Associate Professor John O'Sullivan and other neurologists. This study is currently enrolling participants at multiple trial sites, and I'm sure Dr. Gordon can explain to us more how to get involved. So I will hand over to you for you to talk to us more.
Fantastic. Thank you all for joining us this afternoon. I'll just get my slides up here. So the title of our talk today is Understanding and Treating Microbiome Dysfunction in Parkinson's Disease. So I'm a toxicologist by training. I did my PhD in Parkinson's disease in the US, in Iowa, under the supervision of a renowned neurotoxicologist, Professor Kantasamy. So I've been working on Parkinson's most of my research career for about the last 10 years. So we've mostly focused so far on inflammation and how the inflammasome pathways, for example, contribute to disease progression in Parkinson's. And the microbiome trial that we've started, or that we're hoping to get started in the next few weeks, is an exciting new area for our research group at the University of Queensland, and it's a relatively new area even for my lab group at the UQ Centre for Clinical Research. So this first clinical trial is part of what's called the Queensland Drug Repurposing Initiative, or QDRI, and we've set up QDRI, or we're setting up QDRI with some funding from the Advanced Queensland Initiative from the Queensland Government, where it's essentially clinical trial capacity for drug repurposing in Parkinson's. And we're starting with this as the Combo PD clinical trial as our first trial for the drug repurposing initiative. So our clinical lead, Associate Professor John Sullivan's discussed in depth the benefits of clinical trials and constipation in Parkinson's in the previous talk. So I'm sort of picking up from John's talk a few weeks ago. And we'll be covering how the microbiome and why we're looking at the microbiome as a potential avenue for disease modification in Parkinson's, and how we hope to achieve that, at least in the first stage with this trial, in terms of constipation. So just taking a step back to why we got into this, if you look at some of what's considered the four big questions in Parkinson's, so this depends on who you ask really as to what the biggest challenges are. And we know from talking to patients that everyone's Parkinson's disease is different. Everyone has a different journey. It's a spectrum of different conditions and symptoms. So some of what was written in the journal Nature a while back was just in terms of a few years ago now. But most of these questions are still relevant. We're still understanding this. So how does Parkinson's disease begin? What is the role of alpha-synuclein in Parkinson's, for example? What is the role of the gut microbiome, or the gut itself in Parkinson's and the gut-brain axis? And what is the best way to divide people with Parkinson's disease into different subtypes, into these what we call different Parkinson's diseases, because everyone seems to be different and one single clinical trial or one drug may not work for everyone. So we decided to focus on one of these big questions with this research program because we had a partnership ongoing with industry partner Anagenics and Microba, who are world leaders in microbiome sequencing. So right here in Brisbane, and we had this opportunity where we partnered with Microba, and so we thought we'd attempt to answer some of these questions as to one of these big questions in terms of what is the role of the gut microbiome in Parkinson's. So we're doing a few different things in our lab. We're doing some biomarker research to see if changes in the gut microbiome in Parkinson's can contribute to the disease. And in our Combo PD trial, we're trying to restore the microbiome or some of the microbiome changes in Parkinson's with the hope of potentially trying to alleviate some of these symptoms, like constipation, for example, which is a very early symptom that occurs in the disease course. So if you look at, again, the spectrum and the timeline of Parkinson's disease and the symptoms that occur. So constipation, which is related to gut dysfunction and gastrointestinal dysfunction, is one of the earliest symptoms. It occurs in what's called the prodromal disease phase of Parkinson's. So that's where it's essentially before you diagnosed by your neurologist, or in many cases before you even see your GP. So that's this prodromal phase in Parkinson's can last years to decades in many people. So, and if you look at the symptoms, you've got the motor symptoms here in the light blue color and up there in that spectrum of the disease. So that's typically when people are diagnosed with Parkinson's, when motor symptoms set in. But these non-motor symptoms, things like constipation, depression, changes in sleep, those things occur years to decades before the onset of these motor symptoms. So traditionally, the therapeutic strategy for Parkinson's has been to treat people after they've got these motor symptoms, and we're trying to slow the degenerative process, the loss of these dopamine-producing neurons in Parkinson's. And we've usually gone in with drugs like kinase inhibitors, different strategies like exenatide and things like that. But by and large, those trials have not been very successful. We're still understanding why and learning from all of the unsuccessful clinical trials so far. But an emerging field of thought is that we should look earlier in the disease course, in the disease phase. So even looking at things like constipation and changes in sleep behavior, and even trying to target that, because the earlier we can intervene in the disease, the better our chances are of slowing it down or stopping it or potentially reversing it. At this point, we'll even take slowing down or stopping the disease as a big win. So that's where we're kind of approaching this battle with trying to find new treatments and markers for Parkinson's. We're looking much earlier than the onset of motor symptoms. We're looking here in this prodromal phase, which is potentially people who have constipation and things like sleep dysfunction, with the hope of getting at the disease earlier and then potentially preventing any further deterioration of symptoms. So if you think of the gut microbiome, and it's essentially an ecosystem in the gut. So there are roughly 10 times more bacterial cells than the number of human cells. So that's a very, very large number. It's 10 to the power of 14. The microbial genomic content in the gut microbiome is 100 times more than the human genome. So it took a landmark effort in the early 2000s to sequence the human genome. That's led to a lot of groundbreaking discoveries. But we're still battling with the microbiome genes. We, in fact, are at best our coverage or understanding of the microbiome genes that changes that occur in humans. We can only sequence and identify about 60 to 70% of the genes in the human gut microbiome. So there's still a lot of unknowns, and even with the 70% that we understand, we don't really know. We can now tell what the changes are in the gut microbiome in people that are healthy versus people who have Parkinson's or other diseases, but we really don't know what those changes mean. So if you look at the microbial ecosystem, there's different species of bacteria here that reside in different parts of the gut, the human gut. The colon has the most abundant and the most diverse range of bacterial species, but there's also less bacteria present in the small intestine, and even some in the stomach, where you have stomach acid essentially prevents bacterial growth. And the microbiome, this microbial ecosystem, is affected by age, it's affected by antibiotics, of course, it's affected by stress, it's affected by genetics, where you live, your diet has a huge influence on the microbiome. But we still don't understand what happens with what triggers the changes in the microbiome that occur in Parkinson's. We now know that there are very specific changes that occur in people with Parkinson's and their microbiome, but we don't really know what that means yet, but we're still understanding it. But if you look at some of the biggest factors that influence the microbiome, things like age, for example, exposure to pesticides and chemicals. All of those things have very strong links to Parkinson's. They are known risk factors for Parkinson's, and they also impact the microbiome. So in terms of the influence of microbiome on human health, we're starting to understand, especially over the last decade or so, that the gut microbiome has a profound impact on gut health. And then gut health eventually impacts things like metabolic health, impacts your brain health, cardiovascular health. All of those connections are now being unraveled. So if you take just a simplified approach to how the changes in the gut microbiome impact the host or humans, if you think of it, the gut, there's 10 times more bacterial cells compared to human cells in the gut. So these bacteria produce genes and proteins. We know about 70% of the genes now, and we know some of the proteins that they make. We know some of the metabolites that they make. But the microbiome also, a key role that it plays is maintaining this mucus layer at the epithelial cell lining. So that's the lining of the gut. It's lined by mucus, and having a healthy mucus layer is really important to prevent inflammation and maintain a healthy gut barrier. So in people who have a healthy gut and general health, their microbiomes are, if you sequence the microbiome and try to understand what those bacteria are doing in healthy people, you'll find that their microbiome isn't producing proteins that can degrade this mucus layer. And basically their microbiome is not contributing to inflammation, or maybe even promoting the suppression of inflammation or the resolution of inflammation at the gut-brain axis. So if you take the opposite scenario in diseases like inflammatory bowel disease or even in Parkinson's, the emerging evidence suggests that the gut microbiome has actually changed profoundly in these diseases, and that the kind of profile that's emerging with the changed microbiome in these inflammatory diseases, including Parkinson's, is that the microbiome can promote inflammation. There's the microbiome in these inflammatory diseases can promote the degradation of this healthy mucus layer and this healthy gut barrier. And all of that contributes to what's called unresolved inflammation or ongoing inflammation, which actually we knew even decades ago in Parkinson's. Unresolved or neuroinflammation is something that's been strongly linked to people with Parkinson's. People who take anti-inflammatory drugs, for example, in their second, third, fourth decade of life have a reduced risk of Parkinson's. So inflammation and the link to Parkinson's is very strong. The link between gut microbiome and inflammation is something that's just emerging and something we're looking to study in our trial as well. So that's kind of where we're approaching our therapeutic strategy with. We're trying to prevent this unresolved inflammation or try and restore this gut microbiome ecosystem in people with Parkinson's, with the hope that it can prevent or slow the disease. In the first stage, we'll be looking at constipation in our particular trial. So this concept of the gut-brain axis in Parkinson's, for many years, we focused on Parkinson's as a brain-centric disease. Everyone focused on restoring these cells, the loss of these dopamine. So sorry about that. We'll pick up here. So again, the concept of just the gut-brain axis in people with Parkinson's. For decades now, we've looked at Parkinson's as a brain-centric disease. The majority of our therapeutic strategies and treatments have been focused on the brain and trying to stop this neurodegeneration. Increasingly, over the last 5 years, we found these changes in the microbiome that we've seen line up really well with some of the early symptoms that are linked to people with Parkinson's. So things like gastrointestinal disturbances, changes in gut motility, so that's essentially things like constipation, increased pain, for example, depression and anxiety. All of those have very strong links to changes in the microbiome, for example. So essentially, this concept of Parkinson's disease has shifted. This paradigm has shifted over the last 5 to 10 years, and we're looking at it more as a gut-brain axis rather than just a purely brain-centered disease. There's emerging theories that Parkinson's could be gut first, or there could be forms of Parkinson's which are brain first. All of those have been proposed in the last 5 years by experts based on all of the emerging evidence. We still don't understand all of the details, but that's where our understanding of the disease, a very complex disease, which is Parkinson's, is taking us. So what are the changes in the microbiome with people with Parkinson's? So over the last 5 years or so, there have been a large number of studies in people with Parkinson's in different countries, in Europe, in America, in China, Japan. All of them have shown that they've established now quite consistently that there are changes in the microbiome with people with Parkinson's. And some of these microbiome changes correlate with whether you have a motor phenotype or strong motor symptoms. Some of them correlate, changes in these microbiome bacterial species correlate with how long you've had the disease. And so the emerging consensus is that, yes, there are changes. We still don't understand what all these changes mean. So now we're starting to look at what could we do with the current understanding that we have of these changes in the gut microbiome of people with Parkinson's. So a really interesting paper that came out in Movement Disorders this year did what's called a meta-analysis. So they took all of the previous published literature on the gut microbiome of people with Parkinson's, and they looked for trends in amongst the previous studies that have been done. So they combined the studies from Europe, from America, from Asia, and in people with Parkinson's on the gut microbiome changes. And they wanted to understand which of the changes are consistent, because if you imagine people are diverse in all these different countries, the microbiome itself changes depending on where you live. So this group looked at which are the most consistent changes in people with Parkinson's. And they wanted to understand what those changes could mean. So we'll get into the specific data set of that a bit down the line. And so if you look at the mechanisms by which the microbiome could contribute, or these changes in the microbiome can contribute to Parkinson's, we're still at a very early stage in understanding what these changes mean. But the emerging evidence is obviously people have looked at whether the gut microbiome could influence, for example, Lewy body aggregates forming in the gut. And so what they've done in mouse studies, at least so far, is taken Lewy body aggregates, so these are alpha-synuclein aggregates, and they've injected it in the gut, in the stomach of mice, and they've now traced that these aggregates in the gut can also propagate to the brain, similar to what's been observed by Brack and other people in people with Parkinson's. So in terms of understanding how the changes in the microbiome can contribute to Parkinson's progression, the data supports things like the gut microbiome can cause inflammation, gut inflammation. It can trigger the spread of synuclein. There are some studies which suggest that the microbiome can even impact on synuclein aggregates forming in the gut, in the stomach, and that spreading to the brain. There's, of course, links to the neuroinflammation. And then all of these processes eventually lead to the loss of dopamine-producing cells and the symptoms of Parkinson's. So there's been a lot of studies on vagotomy. So that's essentially if you sever the vagus nerve, you can prevent some of the spread of the disease from the gut to the brain. So you can do those studies in mice quite elegantly. You can sever the vagus nerve and prevent all of this spread. So all of the studies have been done in the last few years, and the evidence is accumulating that Parkinson's could potentially start in the gut with these changes in the microbiome, and some of those changes can lead to the progression of the disease. So this is yet to be proven in humans conclusively. It's even difficult to do those studies in humans, and almost impossible because of the variation and to get consistent numbers. But there is some emerging epidemiological evidence. People who have vagotomies sometimes have a lower risk of Parkinson's later in life. So the evidence is accumulating. So going back to the study from Japan, where they looked at the microbiome changes in Parkinson's, what they found was consistently across, if you take all of the previous studies that were done in people with Parkinson's across different countries, you see these very specific changes in people that are linked to Parkinson's. So there's these specific bacterial species, Akkermansia, Lactobacillus, Bifidobacterium, which are actually increased in Parkinson's. And you see also consistent changes in specific species of bacteria that are decreased in abundance in people who have Parkinson's. And one of the most abundant or most consistent changes is this bacterial species called Faecalibacterium. So that actually represents what they've done here. Faecalibacterium represents about 6 different strains of this bacterial species. There's different subtypes, and there's actually different strains that have different functions in limiting inflammation. So you find these bacteria that are involved in suppressing inflammation, maintaining a healthy gut barrier, and maintaining a mucus barrier in the gut are all decreased in people with Parkinson's. So we still don't understand, again, what this means in terms of propagating the disease or progression of the disease. And that's what we're trying to understand with our current research efforts all around the world. So what we've decided to focus for our combo clinical trial is focus on trying to restore some of these changes that we've seen in Parkinson's disease. So we teamed up with a company called Anagenics and another company in Brisbane, Microba, who are world leaders in microbiome sequencing. And we've taken a therapeutic strategy to see if we can increase the levels of Faecalibacterium in people with Parkinson's. And we're trying to see if by doing that, whether we can restore healthy gut function or promote healthy gut function, and if that has any impact on the gastrointestinal function, starting, of course, with constipation, because that's relatively easy to measure. So John O'Sullivan's talk a few weeks ago covered the details of constipation and what are the specific measurements that we're doing in the clinic. So anyone looking to get more details on that can look at John's talk. I'm going to focus more on the microbiome side of things and the research side of things. So in terms of FPRAU and Faecalibacterium, so what we're looking to do, so our study is called the Combo PD study. So it's a double-blind study, and there's a placebo-controlled group, and it's a crossover trial, which means everyone who participates in the trial will undergo treatment with the prebiotic and the placebo. So, and it's the first study. We consider this to be a first study looking at constipation. We're not looking to see if it slows down the progression of the disease, because of the timeline that we have and the costs involved. So we're looking at specifically at constipation and the microbiome function in the gut for this particular trial. And our hope is, if this study is successful, if we see that there's improvement in constipation, if the Faecalibacterium levels in people with Parkinson's are restored, and there's potential evidence of that in the biomarkers that we look at, then potentially that gives us hope to go for another trial, a larger trial on the back of this one. So they call the combo study. We've named it the combo study because it focuses on constipation and the microbiome. And our aims of our study are to determine the efficacy of this prebiotic intervention, so Livaux, for the treatment of functional constipation in Parkinson's. So that's our main aim in the clinic. That's what we'll be measuring. That's something that John's covered in detail in his last talk. And on the research side of things, in our labs at UKCCR, what we'd be doing is to determine if treatment with Livaux, if treating the microbiome with this prebiotic, can increase the relative abundance of FPRAU-SNTSI. So if that can increase the levels of this microbiome, this bacterial species, FPRAU, that's lost in people with Parkinson's. And then we're also looking at what the microbiome is producing in people with Parkinson's. And if any of that changes, we're looking at things like inflammation, markers of leaky gut, for example, in people with Parkinson's, and signs that there is of the gut-brain barrier, for example, the gut barrier, the mucus production, and things like that. So we'll be looking at markers of what these microbes are making, whether there is improvements in the microbiome metabolites that can suppress inflammation. And we're looking at also microbiome metabolism in all of the people with Parkinson's when they're on the placebo and when they're on this a prebiotic called Livaux, which we know restores the levels of this F-Prausnitzii in people with Parkinson's. So our study design, you have initially a screening period, and then you will be on the prebiotic treatment, which is aimed at restoring F-Prau in people with Parkinson's. And so that's for a period of 6 weeks. We then have a washout period when you cross over. So if you were on the placebo group, you will then cross over to the treatment group. And if you were on the treatment group, you will go back to you go to the placebo group. And then we want to see and for a period of 6 weeks. So the washout period here is 4 weeks, and then you switch over. So all participants will receive the treatment and the placebo. So unlike some other trials where you can get either one, we've designed a study so that all participants will benefit from the treatment. And the washout period allows us to switch over. And one of the other benefits of participating in the study is you'll get a very detailed microbiome profile from the start and the finish and the end of the study. And that's through our industry partner for the trial, Microbiome. So all of the participants will get an idea of what their microbiome looks like. And I know that's of great interest to most of the people with Parkinson's that we've talked to. So that's another benefit of participating in this study. So why are we choosing this prebiotic approach to restore F-Prau or Faecalibacterium prausnitzii? So the reason we do this, I know most people, when they think of the microbiome and targeting the microbiome and improving the microbiome, the go-to approach is to take a probiotic or a live bacteria or frozen bacteria, for example, in a pill. It's easy, it's quick, and it's simple. There's been some evidence of that. So the problem we have with F-Prau is that F-Prau, the one that's lost in people with Parkinson's, it belongs to a class of bacteria called anaerobic bacteria. So these bacteria only grow in the absence of oxygen, which is in the intestines. So these bacteria proliferate, they increase in abundance only in anaerobic environments. So when they get in contact with oxygen, for example, and you try to make it in a tablet or even in the stomach, this bacteria won't survive. So F-Prau is only present in sites in the gut that are anaerobic or which are devoid of oxygen. So it's not really suitable for formulating it as a probiotic or a live strain. It's not like Yakult, which you can just take as a liquid. So we've taken the prebiotic approach. So what we're doing instead is we partnered with this company called Energenics, and they've got a proprietary substrate for F-Prau. So they've extracted the food that the Faecalibacterium feeds on, or what it uses to grow in the gut. And we're going to try to restore the levels, the F-Prau that's already in the gut of people with Parkinson's. And by providing the specific peptides and oligosaccharides that it feeds on with the aim to restore its relative abundance. So the good thing is this approach has been shown to improve functional constipation in people with Parkinson's. So Energenics has previously published trials, and they've got human data showing that it improves constipation in people who are functionally constipated. So those are healthy people who have constipation, and they go to their GP, and it's called functional constipation. So they're not people with Parkinson's, but they have shown in healthy people who have constipation that this Livaux, which we're testing for in our combo trial, can restore functional constipation and even increase the levels of F-Prau. So you can see here, before and after they were on the prebiotic, you have an almost doubling of the F-Prau population in the human gut of people with functional constipation. So why are we focusing on F-Prau? Again, F-Prau is one of the most consistently reduced bacterial species in people with Parkinson's in their gut microbiomes. In healthy individuals, F-Prau is one of the most abundant bacterial species in the gut microbiome. It ranges anywhere between 8 to 16% of the total gut microbiome population. So it's one of the most abundant. And it's also strongly anti-inflammatory. It promotes healthy gastrointestinal function. And restoring F-Prau, there's some evidence once you restore F-Prau that it also improves the overall bacterial ecosystem. So it's an approach that's being used in a lot of inflammatory diseases. No one's tested this yet in Parkinson's, so we're hoping to be the first ones to try this out. And so in terms of the research that we're doing in the Combo PD trial, we're doing what's called functional metagenomics. So that's in collaboration with Microbiome. So this allows us to understand this allows us to get the signature of each individual's microbiome before and after the intervention. We're doing targeted and global metabolomics. So that basically allows us to find out what the microbiome, what the bacterial species are making, what they're producing in the gut of people with Parkinson's, and when they're on the treatment or the placebo. And we're also looking at inflammatory markers, things like alpha-synuclein, markers of disease, markers of healthy gut health and gut composition. So we're looking at blood and urine markers. So for the study, we'd be collecting a blood sample, a urine sample, and also a fecal sample, because that allows us to identify exactly what these bacterial species are making and secreting and how that could impact the disease. So it not just allows us to profile the changes in the microbiome, but this study will also allow us to understand what these changes in people with Parkinson's could mean with and without when the FPRAO changes with our treatment and the placebo intervention. So essentially, we're answering, just to tie all this up, we're answering one of these four big questions in people with Parkinson's from the perspective of, can we restore a healthy gut microbiome in people with Parkinson's? Does restoring this gut microbiome help with symptoms? And we're looking at constipation for this clinical trial. And in the long term, after this trial, assuming our trial is successful, and for example, if we can restore constipation and FPRAO successfully in people with Parkinson's, could this be a feasible approach to slow or prevent disease progression in the long term? So we won't look at disease progression in this particular study. It's only going to be for 1 year. It's an initial study that will focus on the gut health and microbiome. But if our study is successful, the next step would be, for example, we would go to the Fox Foundation who fund a lot of our research, and we would try and get funding for a bigger study, a larger clinical trial, with the hope of looking at disease progression as the next step. So this is our amazing team. So if anyone wants to be involved in the study, please get in touch. So we've already started enrolling participants. We've got all of our ethics approvals completed. We're enrolling participants across Southeast Queensland and our clinical sites in both this year, and it'll continue into next year. Please contact Helen. Helen's our clinical trial coordinator, Liz Arnold as well. Any of our clinical team you could get in touch with to enroll. I think Helen's already reached out to several participants from PQI, and so we're the Queensland Drug Repurposing Initiative, and I'd like to acknowledge these are our neurologists and the study sites. So we're recruiting at the Royal Brisbane Hospital, and the Centre for Clinical Research is where we're based. So we've also got sites at the Princess Alexandra Hospital, St Andrews, the Gold Coast, and at Wynnum. So please feel free to get in touch. All of our contact information is here. I would like to acknowledge our amazing team who make all of this possible, our funding agencies who support this work. The Queensland government has made a huge investment in this through their Advanced Queensland program. And we've also had funding from Shake It Up Australia and Wesley Medical Research, and our industry partners who are collaborating with us on the study, Anagenics, who's providing the intervention that we're testing, and Microbiome, who are helping us with the microbiome sequencing. Thank you all for your attention.